Hello, students. In this virtual lab, you will be measuring the rate of photosynthesis in an Elodia plant. An Elodia is an aquatic plant. So in order to do this, you do have to click on the link that's provided. This will take you to the lab simulator. In this lab, you are going to measure the rate of photosynthesis. Now, bubbles are going to be given off by the plant through photosynthesis. By measuring the rate at which bubbles are produced, it is possible to tell how fast a plant is photosynthesizing. So it is really important for you to read and follow the directions on how to use this simulator. You will press start and record the bubbles per minute for each of the following light distances. Then you're going to graph your data. Be sure to label your axes, give units, use appropriate intervals, and give a descriptive title. Remember to answer the concluding questions. So what I want you to do is look at the data. This is, you are going to measure the light distance, and you have 5 different variables, and you are going to measure the amount of bubbles per minute that are going to be produced by this aquatic plant. So go ahead and click on open the link. This is going to take you to the page. Now, once you open, it's going to ask you alternate HTML content. It's going to ask you that you do required Adobe Flash pre-layer. So go ahead and click on the blue words that says Get Flash. Click on Allow. It's kind of like a little pop-up. You are going to wait a few seconds for this to load up. Now, what I want you to do is you're going to click on instructions. By clicking on instructions, you're going to have, it's going to give you different arrows pointing to the items on this lab. So measuring the rate of photosynthesis of the Elodia. On the left side, you have a sample pondweed. At the very bottom, you're going to notice a ruler, and this is going to measure the distance between the light and the sample. These are the 5 variables you will move using the slider. You're going to drag the slider at different light distances. Okay, now if I go back, you have 5 distances. You're going to begin with 200 centimeters, 180 centimeters, 150, followed by 120, followed by 100. Okay, at each distance, you're going to record how many bubbles. And by recording, you're going to count. So at this time, I'm going to get the slider and I'm going to move to 200. I'm going to click the start button. Now, once you click on the start button, you're going to notice that there's a plant in the middle. This is the Elodia, and you're going to see a little bubble right in the middle. Okay, you're going to measure that bubble. So bubbles are going to be given off by the plant through photosynthesis. By measuring the rate at which the bubbles are produced, it is possible to tell how fast the plant is photosynthesizing. Now, in order to measure the bubbles per minute, you're going to use the clock that's on the right-hand side, and you're going to click on the little tab that says counter. That's going to give you a plus sign, minus sign, reset. This is going to be a kind of like a clicker or counter. And by clicking the plus sign, it will keep record of how many bubbles you are counting. So I'm going to go ahead and click on the reset. Now, you're going to reset the clock. And what I want you to do is you're going to look at the bubbles inside. Notice how there's 1 and there's 2. I want you to glance at the clock every once in a while so you can record. Now, if you forget one, you skip one, that's fine. It's not a big of a deal. So you're going to go ahead and record until your handle does 1 complete turn. Make sure you look at the middle part. You're going to notice a bubble. Do glance at the timer. Okay, there's one full turn. So I'm gonna go ahead and stop right here and notice how it counted 20. So you're gonna click on full screen, minimize that, go to the lab. So at 200 centimeters light distance, I've collected 20 bubbles per minute. Now you're gonna go back to the lab and the next variable is 180. So you're gonna go back and you're gonna click on the slider and you're gonna move to 180. You're gonna click on start and you're gonna repeat the process all over again. So make sure you're on the counter, reset your clock and begin the counting. There's 1, 2, 3, 4. So you're gonna go ahead and continue until there's a full turn. I'm gonna exit full screen. Once you get that number, you're gonna add it to the next one. You're gonna continue for the 150, 120, and 100 light distance. Okay, as soon as you're done, let's pretend I've collected my data, my numbers, and I'm just making up a few numbers. So with this information, with this data, you're gonna turn it into a graph. So in order to do so, I want you to go ahead and highlight, select your 2 columns, all of your cells, the whole entire table. You're gonna go ahead and right click on copy or use a shortcut, Control C for copy. You're gonna open a new tab, and because I'm already signed up to Google Drive, I'm gonna be able to look at the little settings, the little waffle, and you're gonna find Docs. You're gonna click on it, you're gonna open it and open a new document. So click on the plus, blank document. Okay, excuse me guys, I do apologize. I did get the wrong one. You're gonna open a new tab and you are going to open Sheets. Sorry about that, was my mistake. Go ahead and click on the plus sign. You want a blank one. Now I want to go ahead and click on any cell. Right click and you're going to click on paste or Control V. That data, you're going to select it, highlight. Only that data, you're going to click on insert. You're going to click on chart. So what I want you to do is you're going to provide a line graph using that data. So you're gonna go ahead and let it load. On the right hand side, you're gonna see setup. I wanna make sure it's a line graph. So you're gonna select the one next to it. Now, every graph needs to make sure it talks to you. What do I mean by talks? And that's an acronym. Okay, T stands for title. You need to make sure that your graph has a title. A stands for axes. Your y-axis and your x-axis, okay, needs to be determined, and these axes need to be labeled. L stands for label. The next one, K, stands for key, only if it's appropriate, a legend. S is going to stand for scale. This means what number are you counting? Because you're doing this online, so it's a little bit easier for you. It's already set up for you. You have bubbles per minute on the left side, light distance, and this is what you want to have it. Okay, now the item that is on the x-axis, this is going to be your independent axis or your independent variable. Sorry, it's not axis, it's variable. Okay, and this goes by the acronym of mix, where M stands for the variable that gets manipulated. I, it is the independent variable, and X goes on the x-axis. Okay, this is usually what stands alone what you're manipulating. So at the beginning, this lab, I gave you 5 light distances. So this is considered your independent variable. Now your y-axis is going to be your dependent variable. This is going to use a different acronym. I want you to remember the acronym goes by DRY. D stands for dependent variable. R is going to be the responding variable, and Y, it goes on the y-axis. Now, the responding variable is something that you measure. In this case, in this lab, you are measuring the amount of bubbles per minute at different light distances. Now, that's already set up. Okay, now the only thing that I do want you to change is make sure you add an appropriate title. You're gonna double click, and the best title will be the title that describes what you are illustrating, what you've gathered. So in this case, it is going to be the rate of photosynthesis in Elodia. Okay, and it can be something as easy as that. Now, once you're done, okay, I'm gonna go ahead and label. It could label or title this Google Sheet. You're gonna go to the document where you have the lab. Underneath graph, make sure you've selected, you have your little cursor. You're gonna click on insert, click on chart, click on from sheets, and you're gonna, since it's the most recent one, it's gonna be the first one. Click insert, select. You're gonna have to click on this item and click import. So this is gonna give you the graph. So what you're gonna do is you're gonna make sure you shrink it up a little bit, so it is in place. There you go. Okay, now at the end, I do want you to answer the following concluding questions. Number one, what are the bubbles that you are measuring? What do they represent? Okay, it's a bubble, and if we go back, okay, this is measuring the rate of photosynthesis. Okay, so keep in mind, what are the reactants in photosynthesis? What are the products? Okay, that bubble is a product of photosynthesis. Okay, so think about that. The second question is, based on the results, explain how light intensity affects the rate of photosynthesis. You're going to use your data to help explain your answer. So this is your data. Now let me go ahead and show you. If I were to measure the 100, look at what happens the moment I click start. What do you notice about the bubbles? Okay, are they more frequent? Are they going faster, less frequent, or slower? Okay, versus moving the light further away, there's less intensity. If I start, this is where I started. Look at the bubbles. Okay. So at this time, make sure you do answer the questions. You're gonna close your file as soon as you're done. Make sure before you close, make sure you add your name. Close it. You're gonna go back to the assignment. Make sure you check that the information is there, or if you need to upload your file. And that's it, guys. Hope this helps you.